Good day, everyone. So we will be talking about enzymes. For today, we'll be talking about four things. Number one, 6 enzyme classifications and how we name enzymes. Number two is the 2 types of model enzymes that will be discussed by Miss Riza. Then, we will also be discussing what are the 4 types of enzyme specificity. Then for the last one is, with all of this, how do we regulate all these enzymes? So, before diving on to all of those, we have to first discuss what is an enzyme. Okay, an enzyme is actually a protein that acts as a catalyst for a biochemical reaction. Now, what do we mean by catalyst? We mean by catalyst is to speeding up something that would have otherwise taken so much time. Now, enzymes are globular proteins, and all enzymes are proteins. So what does this mean? Enzymes exhibit the same characteristics of what you would expect from a protein. So, enzymes undergo all the reactions of proteins, including denaturation. Now, for the next slide, it will be the 2 general structural classes of proteins. Number 1 is the simple enzyme, which is composed only of protein, and number 2 is the conjugated enzyme. Now, there are 3 terms that you need to remember with conjugated enzyme. When we say apoenzyme, it is the protein part of the conjugated enzyme. When we say cofactor, it is the non-protein part of the conjugated enzyme. And lastly, those 2 things combined is what we call the holo enzyme. It is basically just a combined apoenzyme and cofactor entity. So that it is for the 2 general structures. Now we'll be moving on to the nomenclature. So let's define. There is an enzyme. What is it? Who is the partner of that enzyme? It's actually called a substrate. The substrate is a thing that binds to the enzyme. So it is the reactant in the enzyme catalyzed reaction. It is the substance upon which the enzyme acts on. There are 6 ways of naming enzymes. But before that, let's remember 3 important aspects of the enzyme naming process. Now, for the first one, we could usually define, or you can usually identify that something is an enzyme if first it ends with an ase. It's almost a giveaway that if something ends with an ase, that it is an enzyme. Now, for the pioneer enzymes, when we first discovered them, it was usually in a suffix in the form of IN. So, remember just those 2 things, ASE and IN. You can have an idea that these are going to be proteins. Next is, usually when we name enzymes, we usually include the substrate and as well as the type of reaction that it goes with. So scientists usually do this to identify a enzyme easier. So we include the substrate and the type of reaction that it does. Now we just put an ase on the end. Now, for the 6 enzyme classification, here we go. Let's go to the first one. First one is oxidoreductase. It is an enzyme that catalyzes an oxidation reduction reaction. Then for the next one, it is transferase, which is usually just transferring a functional group from one molecule to another. That's it. It's just that. Number 3 is hydrolase. We have all learned hydrolysis reaction in our geochemistry, where we learned that hydrolysis is just addition of a water molecule to a bond to break it. The number 4 is lyase. Lyase is an enzyme that catalyzes the addition of a group to a double bond or the removal of a group to form a double bond in a manner that does not involve hydrolysis or the oxidation. That is number 4. Number 5 is isomerase. Isomerase is just from the word isomer. It is literally just rearranging the atoms in a substrate. Number 6 is ligase. It is the enzyme that catalyzes the bonding together of two molecules into one with the participation of ATP. So this needs energy. So enzymes are in the form of ligase is something that makes two substrates or two molecules bind together. So that's just ligase. So those are the 6 major classifications of our enzymes, and here is actually a table of where we can find all of these examples. Please take a picture, pause the video and take a picture and search these for yourself and where they fit. You'll be amazed of how incredible and how useful our enzymes could be. Moving on to enzyme active site, but this is going to be discussed by our Miss Riza and Mendoza. Riza, please take the floor.
Thank you, Mr. Ramos. So now let us now move on to the enzyme active site. So it is a relatively small part of an enzyme structure that is actually involved in catalysis. So the active site in an enzyme is a three-dimensional entity formed by groups that come from different parts of the protein chains. So these groups are brought together by folding and bending, which are the secondary and tertiary structure. So the active site is usually a crevice-like location in the enzyme. So next is the enzyme substrate complex. So it is the intermediate reaction species that is formed when a substrate binds to the active site of an enzyme. So in enzyme-controlled reactions, this alternative pathway involves the formation of an enzyme substrate complex as an intermediate species in the reaction. So there are 2 models for substrate binding to enzyme. The first one is the lock and key model. So to account for the particular way an enzyme recognizes a substrate and binds it to the active site. The active site in the enzyme has a fixed, rigid, and geometrical conformation. Only substrates with a complementary geometry can be accommodated at such a site as much as lock accepts only a certain key. So in the lock and key model, the enzyme has a predetermined shape for the active site. Therefore, only substrate whose shape and chemical nature are complementary to those active site can interact with the enzyme. So here is the diagram of the lock and key model. So the next type of model is induced fit model. So the induced fit model allows for small changes in the shape or geometry of the active site of an enzyme to accommodate a substrate. So a good example is the changes that occur in the shape of a glove when a hand is inserted into it. So the induced fit is a result of enzyme's flexibility. It adapts to accept the incoming substrate. So here, substrate contact with the enzyme will change the shape of the active site. The enzyme active site, although not exactly complementary in the shape to that of substrate, is flexible enough that it can adapt to the shape of the substrate. So here is the diagram of the induced fit model. Okay, so now let's move on to the enzyme specificity, which will now be reported by Ms. Sabile.
Thank you, Riza. And now we will move on to the enzyme specificity. But first, let us discuss its meaning. Enzyme specificity is the extent to which an enzyme's activity is restricted to a specific substrate, a specific group of substrates, a specific type of chemical bond, or a specific type of chemical reaction. The degree of enzyme specificity is determined by the active site. Some active sites accommodate only one particular compound, whereas others can accommodate a family or closely related compounds. So now, we will discuss the 4 types of enzyme specificity. The first one is absolute specificity. The enzyme will catalyze only one reaction. Catalase is an enzyme with absolute specificity. It catalyzes the conversion of hydrogen peroxide to oxygen and water. The second one is group specificity. From the word itself, group, the enzyme will only act on molecules that have a specific functional group, such as hydroxyl, amino, or phosphate groups. Carboxypeptidase is a group specific. It cleaves amino acids, one at a time, from the carboxyl end of a peptide chain. The third one, linkage specificity. The enzyme will act on a particular type of chemical bond, irrespective of the rest of the molecular structure. Phosphatases hydrolyze phosphate ester bonds in all types of phosphate esters. Linkage specificity is the most general of common specificities. And lastly, stereochemical specificity. The enzyme will act on a particular stereoisomer. Chirality is inherent in an enzyme active site because amino acids are chiral compounds. An L-amino acid oxidase will catalyze the oxidation of the L form of an amino acid but not the P form of the same amino acid. And for the next and the last topic, the enzyme inhibition will be discussed by Hadriel Ramos.
Alright, so thank you, Angelica, for that wonderful discussion. We will be next tackling about our enzyme inhibitions. So, enzymes are very powerful. So, it is only natural that our body does something to regulate them. Now, one of the ways that we can regulate enzyme is to actually inhibit it. Now, how do we actually inhibit this? There are three types. For the first type, it is the reversible competitive inhibition. It is where we create a substrate, or we create a substrate that is like the substrate of the enzyme, and we let them compete. So it's basically the molecule that sufficiently resembles an enzyme substrate in shape and charge that it can compete with the substrate or occupancy of the enzyme's active site. So when they compete, the activity of that enzyme will be reduced. Now, why do we call them reversible? It's reversible because these things work in majority of who's ever in the amount, in better amount or greater amount, will be the one that will bind to the enzyme much more. So we call this reversible competitive inhibition. Now, how about the second one? How does it actually differ from the first one? This one, the substrate that we create is not actually a substrate that binds to the active site, but somehow other than the active site. And this makes it difficult for the enzyme to move. Now, how do I make this to a real life example? Just imagine this is like putting a clamp to your car's wheel. You're not actually blocking the keyhole, but you are immobilizing by binding to the wheel. So that's how I think of it. Reversible non-competitive inhibition. A molecule that decreases enzyme activity by binding to a site other than the active site. Then the next one is irreversible inhibition. Now, how does this differ to the first one? It actually differs to the first one in one very simple principle. The only difference is the intermolecular force between the substrate and the enzyme present. So basically, when it tries to cleave on, when the substrate tries to cleave onto the enzyme, it does not let go. So the original or the substrate that it actually has to bind with cannot bind because there's already a substrate that is already there that won't just bud off. So that is the irreversible inhibition. Now, why do we regulate enzyme activity? There are actually two very good reasons. Number one is the substrate is low. So we don't actually need enzymes when the substrate concentration is low. We're just wasting energy if we do that. Number two is that the product of that enzyme, like what that enzyme does, is already too much, and we can actually see this in glucose pathways when there's enough activity, or if there's enough ATP, if there's enough energy, enough products, that we don't need those enzymes anymore. So we're just really wasting energy, and we just need to turn them off. Now, how do we turn them off? Besides what I have just mentioned earlier by inhibition, there are three mechanisms. Number one is the feedback control with allosteric enzymes. Now, allosteric enzymes is just a very fancy word to describe something simple. Allosteric enzymes are enzymes that has an active site and another site for inhibition. So that's literally just it. Two binding sites, one is for the substrate and the other one is for regulation. Number two is proteolytic enzymes and zymogens. I think you can find these in the form of fibrinogen turning into fibrin, or plasminogen turning into plasmin. So these enzymes are really just like they are precursor enzymes that turn into full mature enzymes. How do you call it? Mature enzymes. So yeah, precursor to the main enzyme. Then third one is the covalent modification. In overview, that is just literally the adding of a phosphate group, or adding of a phosphate group to make the enzyme difficult, or to do its activity. Actually, it can activate or inhibit an activity. So let's go to the allosteric enzymes. Just what I said. These are enzymes that have first, let's define their characteristics. They are protein chains, quaternary structure, and they have two kinds of binding sites. Just what I said, substrate and regulator. The next slide. So binding of a molecule at the regulatory site causes change in the overall three-dimensional structure of the enzyme, including structural changes at the active site. So whenever a substrate binds, or a molecule binds to the regulatory site, the enzyme actually changes in shape, so the substrate won't enter into the active site, thus changing its activity. Next slide, the feedback control. That's how the whole process is done. Next slide is what I mentioned earlier. A zymogen is the inactive precursor of a proteolytic enzyme. So these are just enzymes that are precursors to the active form. Next is, so here's a diagram. The next is just the covalent modification of enzymes, and usually it's just phosphorylation. What I've said, phosphorylation can either activate or inhibit enzyme activity. It just depends on which enzyme we're talking about. So phosphorylation, it can either activate or inhibit. So that it is all for all the types of enzymes that we have discussed. And here I am with Angelica and with Risa. Thank you so much for listening, and we'll see you in the next one.